And Diane, if you want to go ahead and share your screen, we can get started too. Well, actually, never mind. Go ahead, Justin. You're good to go.
Yeah.
Thanks, Thad. Good afternoon, everyone. It's my pleasure to introduce our Ecology Center speaker, Dr. Diana Six. As we'll see over today and tomorrow research seminars, Dr. Six's research focuses on bark beetles as systems to understand both the evolution and maintenance of ectosymbioses and the feedbacks between disturbance agents and forest adaptation. Given the systems focus, Dr. Six's work ranges in scale from microbes to landscapes and occurs across multiple ecosystems of North America, Europe, and Southern Africa. Dr. Six is currently professor of entomology and pathology at the University of Montana in Missoula. She's also an extraordinary professor of ecology at the University of Pretoria, South Africa, and a fellow of the Royal Entomological Society. In 2018, she was presented with the E.O. Wilson Biodiversity Pioneer Award for her research in bark beetle ecology and climate adaptation of forests. Please join me in providing a warm virtual welcome to Dr. Diana Six.
All right. Okay. I'm gonna attempt to share my screen now. Hopefully people can see it. Is that showing up?
Yes.
Everybody's muted, so I'll have to. Okay, thank you. Thanks for the invitation, and thanks for coming to hear my seminar. It's really nice. I can't say nice to be here because I'm not in Utah, I'm in Montana. But it's actually very lucky we have Zoom, because if I was supposed to be there in person, our airport has been shut most of the last 3 days, so I wouldn't have made it. So at least with Zoom, I get to participate. Today in my first seminar, I'm going to talk about some work we've done to try and answer a question that people have been asking for literally decades, and that is, do fungi matter with bark beetles? Some people consider them to be more or less hitchhikers, not really playing any important role. Many people have thought that these fungi are pathogens that the insects use to kill the trees. That's pretty much been debunked. But if that's not the case, what kind of roles might these fungi be playing? So today I'm going to talk about the ecological stoichiometry of bark beetle fungus symbioses. And I think probably a really good place to start is to define to people what ecological stoichiometry is. If you think back to when you took chemistry, there was this stoichiometry of reactions that you always had to work out. It was basically the law of mass balance. Whatever goes into a reaction has to come out on the other end in the same amounts. It might be in a different form, but the same amount of elements. And what ecological stoichiometry does is it tracks the flow of elements in this way through ecosystems. And the two people that pretty much developed this area of study are Bob Sterner and Jim Elser. And a few years ago, we were lucky enough to hire Jim Elser to run our biostation at Flathead Lake. And so when he arrived, I began to work with him a bit. And pretty soon we were applying this approach to bark beetle fungus symbioses. And so I'm going to share with you today some of the things that we've been finding. Let's start with some of the work that we did with Western pine beetle. This is Dendroctonus brevicomus. This is an aggressive beetle. It's a tree killer, and it's a specialist on ponderosa pine. It's a really strange bark beetle because most bark beetles will develop in the phloem layer right under the bark, but this beetle actually develops in this really dry, nutrient deficient outer bark. And so it has a really strange biology. So it will develop in these feeding chambers in that outer bark, but it really doesn't feed on the bark much itself. What it feeds on is this layer of fungi that forms in these chambers. And here's a kind of a blow up of what that looks like. It's just this huge layer of spores And that provides a lot of, not most of the food for the beetle. Now, when that beetle is ready to leave the tree, it has to be able to transport the fungi to the next tree so that its offspring have fungi as well. So it has these structures that it develops on its prothorax that when it becomes an adult, they kind of act like little fungal suitcases. They pack those spores in, and then when they disperse, they can take the fungi with them. Now, this whole interaction between the tree and the beetle and the fungi can be summed up pretty much with this graphic over here. You can see that between the beetle and the tree, you have this really strong antagonism, okay? Clearly, the beetle is trying to kill the tree, and the tree, of course, doesn't want to be killed, and so it's defending itself and trying to kill the beetle. On the other hand, you have over here a couple of mutualisms. The beetle has two fungi that it depends on for nutrients. We know that. Oops, I don't know what I hit there. We have an ascomycete, which is Ceratocystiopsis brevicomus, and then we have a basidiomycete down here, an Entomocorticium species. And they're both perfectly adequate to help the beetles develop to adulthood, but the lower one, the basidiomycete, is a little bit better, and so you see a thicker arrow. So we wanted to know what was going on in the system of this beetle that grew in some really, really nutrient deficient substrate. So here's a kind of a cartoon of what goes on in these Western pine beetle infested trees. If you look up here in the corner, this is a tree that I peeled all the bark off of. And you can see these areas that are not stained. So it looks like there's no fungi there. But in reality, those areas with no color are completely colonized by these mutualist fungi. And then over here, you have these black patches. And this is a different fungus. This is Ophiostoma minus. It is carried very loosely on the exoskeleton. It's never carried in those specialized pockets, the mycangia. And most of the time, it's actually carried by these mites that are phoretic on the beetle. So it makes these patches in the tree. Interestingly, where the beetles develop, where this black stain is, they tend to just tunnel for a little while as larvae and then die. And so this fungus is really antagonistic to the beetle. Over in this cartoon, you can kind of see what's going on a little bit better. Adults will bore in, enter this phloem layer.
Okay, everybody's back. Downrange is clear. Eyes and ears on. Eyes hot. We make shoot.
Somebody needs to We have them making these tunnels in the phloem. They lay their eggs. And then those larvae, instead of staying in the phloem like any self-respecting bark beetle would normally do, they actually tunnel out into the bark, make these little feeding chambers, and then they develop and emerge as new adults. Over here with the black stain, they do all the same things, but when they get into the bark, they just sort of tunnel for a little while and they never really grow, and then they die. And so it looks like basically they're starving to death. So we wanted to know what was the difference, what was going on over here with the mutualist fungi, how were they influencing nutrients, and then what was happening with this Ophiostoma minus. One of the things we thought was super interesting about this was that this fungus is actually really closely related to the beneficial or mutualist fungi of the mountain pine beetle and a number of other beetles. So why is it so bad for this one when these other beetles like mountain pine beetle depend on it for survival? So we took an ecological stoichiometric approach, and to do that, we took tree cores. We took them out of trees that had no bark beetles, sort of a control to see what the nutrients were like in a tree with no beetles or fungi. And then we took cores out of infested trees. And we took cores through areas that had the mutualist fungi or the black staining antagonist. we divided those tissues into different parts. We had the bark, we had the phloem, and then we had what we called the outer sapwood, which is right here on the outside of the tree towards the phloem. And then this section in here towards the heartwood, which is the inner phloem. And then we also collected adult beetles and larvae. And we took those and we determined what the concentrations of carbon, nitrogen, and phosphorus were in those tissues and insect stages. And from those concentrations, we could calculate a number of ratios that help us understand what's going on in the tree. They're all pretty simple. This one is the trophic spikeometric ratio. It's simply the carbon to nitrogen or the carbon to phosphorus in a particular tree tissue divided by that same ratio in the insect, okay? If you end up with a TSR of 1, it means you have an exact match with those elements in the tree tissues as the beetle needs in its body. Basically, through a bunch of work that Michael Filipiak did in Poland, we can now kind of get an idea of what these numbers mean. And basically, if the TSR is around 4 or bigger, that means that the carbon is increasing in the amount, and so nitrogen and phosphorus become more dilute as that number gets bigger. And so there's a bigger and bigger mismatch between what the beetle needs to grow and what's in its food. Okay, so it would have to feed more to get what it needs. And then if you have a big enough mismatch, it's really impossible for the insect to actually make that up. So what did we find? I'll walk you through these for the different tissues and the different stages. And so we'll start with the bark. And so for control bark, we can see that nitrogen has quite a mismatch. It's 16, almost 17 TSR. So that's pretty big. If we look at the antagonist bark, it's essentially the same. They're both really bad. If you go down to the mutualist bark, this is still not great, but you can see that when the mutualists are present, that alleviates that mismatch for nitrogen for the larvae. If we look down here with the adults, again, the control and the antagonist bark are essentially the same thing. They're both really bad and they're about equal. But if you go to the mutualist, you have this alleviation. Still pretty bad, but at least it's better for a feeding beetle. What about phosphorus? Over here again, the control and the antagonist are both pretty high and they're about equal. They're not very good. But if you go to where the mutualist is present, basically there's as much phosphorus in that mutualist bark as is in the beetle. So this is a perfect match. If we look at the adults, again, the control and the antagonist are a really bad mismatch. But here we have a TSR of 3, which means it's probably very easy for the beetles to get what they need. Now, what about the phloem? Now, I know they don't feed in the phloem, but it was still, I think, really interesting to us to look at the phloem because this is where most bark beetles would normally feed. So we find something pretty different here. The control phloem, again, has a pretty big mismatch, but now it's the antagonist and the mutualist that are similar instead of the control and the antagonist. So here, both the antagonist and the mutualist can reduce that nitrogen mismatch. And again, down here, only the control is really high and both the antagonist and the mutualist can alleviate the nitrogen. And phosphorus is kind of crazy. Here we find a perfect mismatch, pretty much, with phosphorus for the beetles as adults, and with larvae, it's actually an excess. So this kind of makes you wonder, if you're a bark beetle, why would you ever leave the phloem? If with or without a mutualist, it's actually a better place to live. We don't really know, but my guess is that this helps them avoid competition because these trees have a huge number of secondary beetles and other things that pile in that actually could create a lot of competition in the phloem. Now, there's some other things we can look at with the concentrations as well. We can look at what kind of changes occur in those different tissues due to the presence of the fungi. And this again is really simple to calculate. It's just the carbon to nitrogen or carbon to phosphorus in a controlled tissue minus that same ratio in that particular colonized tissue. So if you get a 0, basically the fungi have had no effect. They haven't done anything as far as the elements. If you have a negative value, that means your NRP went down. And if you have a positive, it means that the NRP went up. So this is what we found. So looking at nitrogen, we find that nitrogen went up substantially in the mutualist bark, and then either dropped or stayed pretty much the same in the antagonist bark. This is a pretty small number for these kinds of comparisons. So it's really, really a small change if it happened. Down here in the phloem, we see that nitrogen again goes up. But if you get to the outer sapwood, you can see that nitrogen decreases. And then you can see that it increases towards the center of the tree. And I'll talk about why we think this might be happening. But look over here at phosphorus. This is really a pretty amazing part of the story. When you go to the mutualist bark, look at this value for increase. It's massive. This is a humongous, just gigantic increase in phosphorus when the mutualist is present. And then if you look at where the antagonist is present, it's really tiny. If you go down here in the phloem, there's an increase with the mutualist, a bigger one with the antagonist, but nothing like this. And then if we go down here to the outer sapwood, look at this incredible decrease when the mutualist is present. You know, after all, this stuff has to be coming from somewhere. And it does appear that it's coming from the sapwood. And then the antagonist is also decreasing it, but not as much. But look here in the inner sapwood. This is deep down in the tree, all the way up against the heartwood. When you have the mutualist present, phosphorus basically becomes undetectable. You just can't even find it. It's gone. And even with the antagonist, there's this huge decrease in phosphorus. So essentially, it looks like all that phosphorus is being grabbed up in the center of the tree and moved outward. So this is kind of a way of looking at what we think we're finding. Both of these fungi, the antagonist and the mutualists, are moving phosphorus and nitrogen. They're moving them in similar ways. Nitrogen's depleted here, increases on these two ends. We think that part of the increase here is to support the growth and foraging of the fungi in order to be able to be moving those elements. And phosphorus tends to go one direction. It's being moved from the inside out. So why this difference in effects? Okay, the beetles do really well with one, but not the other. Well, I did some scanning electron microscopy, and this was really informative because it showed that these fungi, that the mutualists, can grow into outer bark. Here's the outer bark cork cells. You can see all these little lumpy things. That's the fungi growing through these cork cells. Looking down here, this is what it looks like when you have the antagonist. The antagonist cannot enter bark, and so essentially it gets stuck, delivering all these nutrients into the phloem. And so you have these little beetles out here. They're right next to all this bounty that's been dropped off in the phloem. But because this fungus can't deliver the goods out to where they're feeding, they basically starve to death. So they're both moving nitrogen and phosphorus, but this one never delivers it to the beetles. So we kind of were wondering, you know, why would the fungi actually do this? It takes a lot of energy to translocate elements through the fungal mycelium. It's energetically expensive. So why are they doing it? Are they doing it to be nice? No. One thing to realize about fungi is fungi are not nice. Fungi are never nice. They might be cute, they might be really interesting, but they're certainly never nice. If they're going to be doing anything for themselves, for anything, they're going to be doing it for themselves. So we know that fungal reproduction is really requires a lot of nitrogen and phosphorus. So we think that they're actually translocating all of these elements, this nitrogen and phosphorus, out to where the beetles are, because that's where they have to reproduce in order to be dispersed. Okay. And we think this kind of developed as a bit of a byproduct mutualism, sort of accidental. This movement of nitrogen and phosphorus may have benefited beetles that were under the bark. And then over time, some of them have evolved and fine-tuned to develop really obligate relationships like we see with this beetle and its fungi. Others not so much. These are not all super tight mutualisms, but some of them are. So there's a few other things you can do with these concentrations. One of those is to develop something called the threshold elemental ratio. And this is the concentration at which limitation of growth, which basically wherever limitation is where there's not enough of a particular element to support growth. And so this is where that switches from one element to another. And so in ecological stoichiometry, usually what people calculate is the carbon to phosphorus ratio for the threshold for the TER. And that's because phosphorus is typically the most limiting element. And since this is a mutualism, we have to kind of think of these associations kind of in a little different way. So if you get a low TER in this case, that means that the mutualist can greatly alleviate the problem of developing in a low phosphorus environment. But on the other hand, if you have a high TER, that means that the mutualists are only able to partially alleviate that low P, that the P is low enough in that environment that even if you have really efficient mutualists, that the host beetle itself is going to have to evolve additional physiological adaptations to deal with that low P situation. So we calculated the TER for the carbon phosphorus ratio by taking the insect carbon phosphorus ratio times the gross growth efficiency for phosphorus divided by the gross growth efficiency for carbon. And typically, gross growth efficiency is determined by some pretty intensive feeding studies where you look at the change in mass depending on how much that organism ingests. Sort of like if I was to eat 15 pizzas, how much would I grow? That's what you do with various organisms and see what efficiency of conversion there is. But these feeding studies are really tough. And it's really impossible with some organisms. Bark beetles, I think, would be a total nightmare to try and figure out the exact GGEs. So there's been a lot of work done in this area with a lot of different organisms, and there's been a lot of work on invertebrates. So we have some estimators that we can use. So we used a gross efficiency for phosphorus of 1 because insects and other invertebrates tend to be extremely good at converting P into body tissues. And then we used a low efficiency for carbon of 0.5 and a high efficiency of about 0.8. And this is what we came up with. We have a low efficiency for larvae of 6672, high efficiency of 4170, a low efficiency of 2816, and a high efficiency of 1760. So what does that mean? Well, trust me, these are screamingly high, okay? They're so high, it almost makes you wonder if there's any phosphorus in this insect, which made us wonder if there's any phosphorus in this insect. how does it match up, or how does it compare to other beetles and invertebrates? So thanks to Angelica Gonzalez, who shared with us her massive data set on invertebrates and their phosphorus contents, we were able to sort of place where our beetle falls. And so if you look here, if you look at the percent phosphorus found in invertebrates, our beetles fall within the lowest 1%. And same with the nitrogen phosphorus ratios, which are often used. We look at insects, again, we're in the lowest 1%. We look at beetles, we're way down there, and actually probably among beetles, the lowest known phosphorus content yet. And so these beetles, even though they have these incredibly efficient mutualists, they go into the tree, they basically deplete every bit of phosphorus from the interior, moving it out to where the beetles are feeding, they still can't alleviate that extremely poor pee environment. And so this beetle has had to evolve a body that is extremely low in pee. So Jim and I kind of went, okay, that was super cool to find that. And that was very interesting to try and tease that apart. But we had the kind of this nagging thing in the back of our minds, made us wonder, well, that's kind of an oddball beetle. What about typical bark beetles? The ones that live and feed on phloem and their fungi, such as you see over here, you know, just like this is like mountain pine beetle, you know, down here in the phloem, they're feeding on their fungi and phloem. What about those? And these beetles are really varied. Some are tree killers, most aren't. Some have mycangia, most don't. And they don't all have specific fungi, some do. So we wanted to take a closer look at some of these other beetles. So these are the ones we've looked at so far. And luckily, these were all in the same kind of tree, the same tree actually that we studied for western pine beetle. And they kind of fill in different parts of the tree. This is up in the top of the tree. Ipsum marginatus fills in where the western pine beetle doesn't attack on the lower part of the tree. And you can see they also have staining. Ips pini has its own specific fungus, an ophiostoma. Ipsum marginatus has its own fungus, another ophiostoma. And then here we have an ophiostoma. This is that antagonist with western pine beetle that was so bad. But when you look at the phloem, what you'll find is there's this little primitive bark beetle, Hylargops reticulatus, that likes to preferentially feed and develop its young in the phloem where this fungus is at. So we took a look at that one as well. And we also looked at this beetle. It's not a bark beetle, but it lives in bark beetle galleries, including all of the bark beetles I just talked about. And this is one of the ips that I talked about we're looking at. It's a lot bigger than this one. This is probably a fifth of the size of one of these tiny ips. So it's a little tiny beetle. And not a lot's known about it. Some people think it's a predator, but no one's ever actually documented it eating anything other than bark beetle poop. So it goes along in these galleries, eats up this frass or bark beetle poop, and that's what it appears to live on. And this intrigued me because bark beetles are already trying to extract every bit of nitrogen and phosphorus they can out of their food. So you wouldn't expect them to excrete very much. You would expect bark beetle poop to pretty much be nitrogen and phosphorus free. Yet this guy is trying to, or is making a living off of it. So we decided to include that beetle as well. I'm not going to take you through all the tables for all these beetles. I'm just going to give you kind of a summary of what we found. And for Ips pini, which is, I should have said this before, but these are all secondaries. They don't kill trees. They come in after something else does, like western. We found with the fungus that's with Ips pini, the symbiont moved nitrogen moderately efficiently and phosphorus very efficiently. Okay, it was really taking the phosphorus out of the tree. Ipsum marginatus, on the other hand, its fungus moved both nitrogen and phosphorus really efficiently. And then Hylargops reticulatus, that one that feeds in the antagonist of Western pine beetle, it turns out that that antagonist, although it does move nitrogen and phosphorus, as you saw before, when you compare it with the fungi of these beetles, it's really inefficient compared to them. And so we have really different kinds of nutrient supplementation, if you will, by these different fungi for their host beetles. So we calculated the threshold elemental ratios for these. And I put Dendroctonus brevicomus down here at the bottom for comparison. But you can see up here for the two Ips species, they have extremely low TERS. They have very high phosphorus contents in their bodies, and they have these incredibly low TERS, whether it's high or low efficiency. But if you look at Hylargops reticulatus, that one that's feeding in that antagonist, it has a really high TER. It's almost as high as you find with Dendroctonus brevicomus. And we're not sure why that is. In part, that fungus is not moving phosphorus very well. But we also know that this beetle feeds on a whole bunch of different fungi, different bark beetles, very opportunistically. So if you're a jack of all trades, you're probably going to have to get by on a whole bunch of different fungi of different efficiencies, and probably have to be able to deal with some of those that are very low efficiency if you're going to survive. So maybe that's why this beetle gets away with being so low phosphorus. What's really interesting is to look at Corticius parallelus. This is our little poop eater. And if you look here, these are freaky, okay? We thought these were high. Look up here. This is insane. These figures are, to date, the lowest known TERS, and also this is probably the lowest known phosphorus in any animal that's been looked at yet to date. There's almost no phosphorus in this insect. And that's crazy, because we need phosphorus for just about everything: ATP, DNA, RNA. If you think about RNA, we need it for ribosomes. If you don't have ribosomes, you can't grow. And that's the main requirement for phosphorus in a cell, is for ribosomes. But this little beetle really is really low. So let me see here. Okay, something else that Sterner and Elster came up with when they put together the book on ecological stoichiometry was a number of hypotheses, and one of those was the growth rate hypothesis, and that is that organisms that have a lot of phosphorus are capable of fast growth and building large bodies, whereas if you don't have much phosphorus, you're going to be slow and build small bodies. So I thought I'd compare the percent phosphorus to the mass in these various insects that we've looked at, those three bark beetles and then the little poop eater, and see if we see this relationship. Luckily, all of these insects have roughly the same generation length, and so we should be able to compare these just using this simple comparison. So here is Ipsum marginatus. This is our biggest beetle by far. It's really a large beetle for a bark beetle, and it's just full of phosphorus. Okay, so that makes sense, that fits. What about down here? We've got Corticius, which is the little poop eater. It has almost no phosphorus, and it also has almost no body. So that fits as well. Over here we have Hylargops, the primitive one that's sort of a jack of all trades. It's low in phosphorus, it's not very big. That sort of fits. But then we have up here is Ips pini. This is really loaded up with phosphorus. but it's not very big. Okay, so why might that be? Well, most of the people that work on ecological stoichiometry really focus super hard on phosphorus, but obviously there's other elements that can be limiting. And we know that the fungus of Ips pini is a really good provider of phosphorus, but it's not very good at delivering nitrogen. And so my guess, and that's all it is at this point, is that maybe nitrogen is limiting in this one and accounts for this smaller size. But that remains to be investigated. Okay, so let me kind of tie up this part of the talk. And basically what we found was that the fungal symbionts of both primary or aggressive beetles and these secondary beetles, this second set that I looked at, the fungi of all of those actually translocated nutrients to where the bark beetles feed. But these fungi varied in their efficiency in delivering nitrogen and phosphorus, and this was reflected in their TER, in their N to P ratios, and to the most part, their adult body size. And maybe the biggest take home here is if you're going to be a beetle living in wood, don't be a poop eater. It's a rough way to go. Okay, I want to end talking about something that I've been working on for some time and then finally published it this year. I'm an ecologist, and so while I really like teasing apart these little symbiotic interactions, it's a super cool thing to study. I also really am interested in knowing what the outcomes of those interactions mean for the greater ecosystem. Okay, and so I've been thinking long and hard about how these symbioses end up influencing kind of the structure and function and composition of forests over time. And what I've done is try to group beetles by their behavior and then how they co-construct the niche within the tree with the fungus to end up with very different outcomes in how, in the intensity or the influence they have on the ecosystem in which they exist. So I'm just going to walk you through this kind of conceptual model that I've developed. And then there's a lot going on here, and I'm not going to spend obviously another hour on this, but I just want to introduce it. And then if you're interested further, you can look up the paper in Current Opinion in Insect Science and get more of the meat. But in general, you can split bark beetles up into 4 main groups. There are the aggressive beetles. These are the ones that mass attack trees, can develop outbreaks that spread even in healthy trees, and so they can build the biggest and the most influential outbreaks. Then we have the aggressive secondaries, which are beetles that can attack large trees or small. They can develop outbreaks, but they're fairly dependent on stressed, weak trees. So whenever those stressed trees are depleted, their outbreaks tend to drop off, and so they don't get as big, they don't have as big impacts. Then we have the non-aggressive bark beetles, and these are secondaries. They go into trees that are either dying, they've been killed by something else. They tend to have more dispersed attack behaviors, so they don't pack into trees as densely. And then we have the parasitic ones that go into live trees, usually only just in individual pairs, and the tree survives, and so they really do act as true parasites. These different behaviors result in different symbiotic development. So you have different types of symbioses with fungi, and consequently different co-construction of the niche. And so if you look at how beetles construct the niche, these beetles that mass attack kill the tree, they provide access for the fungi, their tunneling alters oxygen and moisture content to in ways that help the fungi grow. And then in turn, the fungi provide nutrients, and often detoxify the defenses. This nutrient provisioning is especially important in these particular symbioses with these beetles, because these are mass attack beetles. They go into trees in high densities. So there's not a lot of phloem resource. And so there's the potential for a lot of intraspecific competition. And these beetles tend to make only little tiny galleries and then pupate and then emerge. And so they really don't feed much on the phloem. And that is allowed because they have these fungi delivering nutrients to them. Here we have beetles that also do many of these same things. The fungi do similar things, but they're piling into trees. The densities that they colonize them with is typically lower. They tend to feed more on phloem, and so their dependence on phloem is much higher, and their dependence on the fungi consequently is lower. And then if you go over to these non-aggressive secondaries, they're very dispersed. They tend to be present in the trees in lower densities, and they feed extensively on phloem, and they're less dependent on the fungi. Okay. This all results in these very different dependencies and strengths of these associations with fungi. Okay. Over here with the parasites, you have a really different situation. Okay. The beetles provide access. The fungi are not beneficial. They trigger tree defenses because these beetles are developing in living trees. The trees are continually fighting against these beetles. And if you have fungi, that's going to trigger these defenses even more. And it's been shown that they compete with the larvae for carbon. So the fungi are not beneficial. And in fact, many of these don't carry fungi or just a low diversity suite that is environmentally acquired. There's no specificity. They go in low density, so there's not a lot of any intraspecific competition, and their dependence on phloem is very high or complete. And consequently, we really don't have a symbiosis here, and certainly not a mutualism. Each of these different types of mutualisms or lack of has a very different outcome on the forest. So if you look at these that have these highly efficient mutualists that help them colonize trees in large numbers, you have these massive effects on the ecosystem with these outbreaks. You have moderate effects with these, aggressive secondaries because their outbreaks aren't as big and extensive. They don't reach the same numbers. Non-aggressive beetles are more like slight decomposers. They have very little impact. And then the parasites have basically no real noticeable effect. And so what I've tried to do is take how these organisms interact and then build a model to show how that then can cause these different knock-on effects through an ecosystem. So back to where I started, do fungi matter? I think yes. And so hopefully I have convinced you as well that they do matter. There's clearly a lot of things we still need to study, but more and more we're seeing that they have a very big driving influence on these beetles and their dynamics. With that, I don't know where we are time-wise. I haven't been watching. Looks like we're okay. I'd like to acknowledge Jim Elser, who is my partner in crime with this. He's a limnologist, as you can see by the way he looks at this lake. That's the way he looks at lakes. It's crazy. But he's a phenomenal guy. But even though he's a limnologist, he works on all sorts of things all over the world. So check out his literature. There's a wealth of information there. And also NSF EPSCoR and the Biostation for funding. And with that, I'd be happy to answer any questions you might have.